Hello everyone, in this video we will discuss the question number 13 of GATE 2014. Gram-positive bacteria are generally resistant to complement-mediated lysis because of their thick peptide-glycan layer. Gram-positive bacteria import the membrane attack complex and inactivate it. Membrane attack complex is regulated by the protease produced by the gram-positive bacteria, or gram-positive bacteria cannot activate complement pathway. Which one is the proper statement? We need to check it. So before going that, we will briefly discuss about the complementation or complement mediated lysis of the bacterial cell membrane. So how does it be activated? So first, remember, body antibody detects the bacterial antigen and they will make an antigen antibody complex, right, like here. Then that is recognized by the complement protein C1q. So C1q binds to the antigen antibody complex, and then C1r activates autocatalytic activity of the C1r and it will activate the C1r. So both of them activate C1s. So now this activated C1s, it will cleave C4 and C2, right? So when C1s will cleave C4 and C2, then it will make, after cleavage of the C4, it will make C4a and C4b, and after cleavage of the C2, it will make C2a and C2b. So among them, C4b and C2a, they will come and they will make a complex which is C4b2a, that is also known as C3 convertase, because the C3 convertase hydrolyzes many C3 molecules. C3 is another complement protein, so it will hydrolyzes many C3 molecule, and some of the C3 cleaved product will combine with the C3 convertase to make C5 convertase. So how does it play a role? This C4b2a, which is C3 convertase, so it will bind with C3 and it will cleave C3 to C3a and C3b. Now this C3b will come and it will bind to this C4b2a. So now it will make C4b2a3b, which is C5 convertase, which will use C5 as a substrate. So now at the next step, the C5 convertase will bind to C5 and it will cleave C5 to produce two fragments, C5a and C5b, right? So now this C5, yeah. So now this C5b will bind to C6 and initiating the formation of the membrane attack complex. So now this C5b, which is produced by the cleavage of the C5 by this C5 convertase, this C5b, it will come and it will bind to C6 and C7. So it will make C5b, 6, 7 and at the last C8. So totally it will make C5b, C6, C7 and C8. In total, C5b, 6, 7, 8 complex, which is a membrane attack complex or MAC. So this membrane attacking complex will come and it will bind to the membrane of the bacteria in the bacterial cell membrane, and it will make a pore in the membrane and it will lyse the cell. It will lyse the cell, right? Because it will make the membrane permeable. It will make a hole. So this is the method of bacterial cell lysis by the complement system by recruiting the membrane attacking complex, which is MAC. But some of the bacteria are resistant of this immune system. The example is gram-positive bacteria, because as you know that the gram-positive bacteria has a thick peptide-glycan layer of cell wall. So this thick cell wall, it inhibits or it prevents the insertion of the MAC or membrane attack complex into bacterial membrane. Because of their thick peptidoglycan cell wall layer, they will inhibit the insertion of the MAC into the bacterial cell membrane, right? So that's why it can inhibit this complement mediated cell lysis. So the reason of option A, if you will see, that thick peptidoglycan layer prevents insertion of the membrane attack complex into the inner membrane. So that is the reason because of which the gram-positive bacteria are generally resistant to the complement mediated lysis. So option A is right answer.